Hello everybody, welcome to Chem410 at the University of Toronto. My name is Andrew and I will be a TA for the course. We've put together some videos as kind of a prep or refresher to get you thinking about the various analytical techniques that we will employ in the labs to come. I hope you enjoy. In this video, we are going to be looking at various methods for the quality assurance and quality control. More specifically, spike and recovery, signal to noise, and what that means for our limits of detection and quantification, and then the use of the qualifier ion. Throughout this video, keep in mind that the point of QA and QC is to give confidence to your findings and to convince the reader of your findings that what you claim is correct. It's a way to be completely transparent with what you did and let the reader make their own conclusions as to the thoroughness of the research. So, spike and recovery. What is it and why do we need it? This is a way to probe actually how much of the analyte we can expect to get out of the environmental sample. These spikes should go through the same procedure as actual samples to investigate if and where you lose the target analytes. Now, if you're not familiar with all of the techniques involved with the solid extraction, then I'd recommend going to view that video before moving forward here with the spike and recovery, as I'll be using terminology referencing different steps of a solid extraction. For example, with the house dust samples, we don't necessarily have pure house dust. So when we would normally mix the dust with sodium sulfate, fluorosil, and alumina before loading into the syringe as a cleaning step, we will just mix the fluorosil, sodium sulfate, and alumina. And instead of washing them with clean hexane, we will wash with hexane that is spiked with the organophosphorus flame retardant mixture. This will introduce the analytes to the extraction procedure in a controlled measure, one in which we know the theoretically true concentration, assuming everything goes well and completely extracted. We will compare the experimentally observed concentration to that of this theoretical concentration to determine the extraction recovery. As you can see here, the experimental concentration over theoretical times 100% will give you the recovery. To properly know that the environmental samples have X parts per billion of the analyte, for example, you have to know that the extraction or preparation methods you have employed didn't introduce additional analyte contamination. To determine this, chemists perform blanks. These are pure water or solvent that is treated the same as an environmental sample. For example, is extracted and prepared the same. This will then be analyzed along with the environmental samples to ensure there isn't any background contamination. However, if there is constant background levels of your analytes, which very well could be the case with flame retardants, then these blanks will allow you to determine a baseline, to determine if the sample has more or less than the general background. It is important to mention that in a typical setting, you would perform your QC samples, both spikes and blanks, to assess the quality of the extraction, and then you would include some additional QC samples with your actual environmental samples to ensure extraction quality over time. This leads us to signal to noise ratios. And is what we are detecting actually significant? Is it background to contamination or just instrumental noise? Well, this is why we define the limit of detection, LOD, and limit of quantification, LOQ. These are a way of determining if we can definitively say that the analyte was detected, or we can say that we're able to quantify it. The LOD and LOQ can be calculated in different ways depending on what you're using and what you have available to you. In Chem410, we will use the signal to noise calculated by the instrument and empirically determine the LOD and LOQ from signal to noise ratios of 3 and 10 respectively. To give you an idea of what we're talking about, the instrument has calculated this peak here from my research to be just over 10. If there's any background contamination, however, we will employ a strategy involving taking the mean of the blank's response and add 3 standard deviations of these blanks to calculate the LOD. And then for the LOQ, it will be the mean plus 10 standard deviations. Here, we're trying to understand the limit of the analysis by comparing the signal to the noise of the instrument, or the blanks if there's contamination. Also, keep in mind that this method of blank-defined LOD and LOQ gives values that are quite high, so you really want to make sure you're emphasizing limiting blank or sample contamination if possible, which is the most successful approach in any analysis. So, to the qualifier ion. This is an additional factor to ensure that we are actually measuring the intended analyte. As you know, the second quadrupole from a triple quad, the collision cell, will break up the analytes into product fragments. And based on a chemical's structure, it will break or fragment in different areas, while only one of these fragments will retain the charge from the initial ionization source. Remember that these fragments are measured in a mass to charge ratio, so if there's a neutral loss, it will not be detected. So, if we break up an analyte ABC with, let's say, 20 volts and decide to quantify the product ion A, there will often be a different fragmentation process where if we apply 10 volts, the product ion produced will be BC, or just C. However, potentially not in as high of abundance. Both of these fragmentation pathways will be fairly consistent for the prospective energies applied. Now this is highly reproducible for the sample collision energy, and that is what we will be taking advantage of for this quality assurance technique. The higher abundance, aka the more sensitive ion, will be used as the quantifier ion, and the less abundant, aka less sensitive product ion, will be the qualifier ion. The ratio between these two should remain the same regardless of analyte concentration, if the instrument is tuned properly. Now, why does this help us differentiate between detected chemicals? Well, each chemical should be retained slightly differently on the column. Then, if that doesn't separate them, then the first quadrupole will filter for the correct mass of the parent ion. And then if even that doesn't filter out the interferent, then after the collision cell, the third quadrupole will filter for the specified fragment, for example A in the example I showed you, which if chosen should be entirely specific to your analyte. However, ensuring that the qualifier ion from your molecule ABC is present and in the same ratio as your quantifier ion A, we can be confident that we are measuring the analyte of interest and not the similarly structured interferent. And finally, here's an example from my research. Here, my parent chemical has a mass to charge ratio of 347 and fragments to 185 and 99. Since the 185 fragment has a higher response, as you can see here at the top, 1 to the 8, or 1.33 to the 8, it's going to be my quantifier ion. And the 99, the bottom one, will be my qualifier ion. Now, thanks for listening and participating and taking part in this slideshow. If you have any further questions on any of the QA/QC techniques, please feel free to ask your lab demonstrator or TA. I hope you enjoy the labs.